Assalamu alaikum. I welcome you all in this lecture. Today we are going to discuss about the light reaction for photosynthesis, and before going into this we need to understand two things. Number one is the chloroplast structure and a little bit about the biochemistry or the process. Now the structure of chloroplast over here, don't get upset with it so many labeling. It's pretty much simple. The outer membrane, inner membrane, and the space between them is the intermembrane space. So outer membrane, inner membrane, it's pretty straightforward. Then it contains thylakoids. The thylakoids are the structures which are once arranged in the form of a column. They are called granum, and the space between these granums is called stroma. So the stroma contains ribosomes as well as the chloroplast DNA. From here the concept of evolutionary biologists says that these organelles were actually many million years ago were acting as an independent organism. Later on, once they accumulate inside the organism, they define their own role for the generation of food, while rest of the material exchange and excretions were being designed by the cell itself. So just another thought, but these are the structures which are of prime concern today. Now the yellow things what you are focusing over here are the thylakoid membranes. The photosynthesis begins in these thylakoid membranes. Now the end products which are made, so the light reaction take place in thylakoid membranes. The end products too are made. Number one is the ATP and the formation of NADPH. These are the two core elements which are being produced during light reaction on photosynthesis. But before going into the reaction details, we need to understand two things, that the clustering of pigments. Now these pigments are generally called chlorophylls, and the chlorophylls are 5 in number. These are the photosystem 1 and photosystem 1 and photosystem 2. Now the nomenclature is photosystem 1 is because it was identified earlier, but in fact it is the photosystem 2 from where the system light falls and the process initiates. Okay, so we need to remember photosystem 2 is the process step 1, and photosystem 1 was identified earlier. That's why it is called photosystem 1. Now these photosystems are basically the clusters of chlorophyll, A in more in number. Basically there are 5 chloras, A, B, C and D, and then there is a bacterial chlorophyll present in these photosynthetic organisms. But in plants, majority of the chlorophyll is chlorophyll A. So the clustering of the pigment chlorophyll A in bracket with different proteins form photosystems. Now in thylakoid membranes, we need to understand few basic things, and I am trying to make it as easy as possible. So initially the outer membrane is called stroma, and then there is the inside layer which is called as lumen. Lumen and stroma. Okay, now this stromal mass contains hydrogen ions outside them. Now once the process begins, and this is the step 1, so over here when the process begins, there are multiple chlorophylls are present over here in this photosystem, and these photosystem contain these chlorophyll A. Now once the photon hits these chlorophyll, their electron get excited. So once the electron get excited, let me label an electron in any chlorophyll over here as red color. So just this, let's say this is a chlorophyll and this electron gets excited. Now it communicates this excitation, pass it on to the next chlorophyll electron in the vicinity, and this communicates this message to another one, and the whole cascade begins. Okay, now this message is being communicated at different forums, and ultimately an excited electron is present in the photosystem too. Now this excitation is communicated further down, and with the passage of this excitation communication, it is obviously this excited state is reverting back to normal state. And during this transition, the outer hydrogen ions try to get inside the lumen. Now there is an increased density of hydrogen ion concentration present over here, and the concentration of hydrogen ions inside the lumen is getting higher. Simultaneously, once the photons hit the photosystem 1, the excited electrons, the two electrons are excited at this step. Two electrons are excited at this step. Now these two electrons, along with another shot of photon hitting the chlorophyll A present in the photosystem 1, again excites these electrons, and the electrons once excited tries to reduce NADP to NADPH. Okay, now this is the product we need in light reaction, and that is the product. So over here there is a reduction of NADP to NADPH. So in order to remember NADP to NADPH formation, we coined a term reduction. So reduction means gain. So if we remember this word reg, it means gain of, gain of electron. Let me show gain of electron and hydrogen. Okay, so the gaining of electron and hydrogen is called reduction, and opposite is called oxidation. Okay, now simultaneously there is an increased influx of protons or hydrogen ions present inside the thylakoid lumen, but the lumen doesn't hold these protons free hydrogen ions for long. These hydrogen ions have a natural tendency to revert back into stroma. So in order to go back, they use a channel, and during this channel ATP synthetase enzyme, so this is an enzyme, it gets activated. Now this ATP synthetase is responsible for the formation of ATP. So here comes the two components, 1 and 2, both are being formed. Now let's bisect this whole process into an easy formation so that we can get an understanding what's going on. Simultaneously I am trying to delete this portion so that you can get a clear understanding and don't get confused. So light hits on the photocell system 1, 2 electrons get released. Now these electrons are released from here, and the message is being communicated to the step 2 that high energy electron excite reduce the energy during flow. So since the energy of that electron is gradually declining during the whole process, so the hydrogen influx from stroma to the lumen is being observed at this portion at step 2. Number 3 step is that the photon hits again in the photosystem 1, and this hitting results in an activation of these electrons, and these electrons excited get into the reduction of NADP to NADPH, right? And this NADP to NADPH results in the formation of a product which is required in dark systems, dark reaction system. Now the third step is that this thylakoid lumen contains higher concentration of hydrogen proton. They want to exit, so they exit using ATP synthetase activation. Now this ATP synthetase is mainly responsible for the addition of pyrophosphate to ADP, resulting in the accumulation of ATP. So here comes an interesting question, and that question is basically a key to answer one more puzzle. That is, what about the initial electron excited? The two electrons, as I mentioned, are excited at this point. Now if the electron is excited and is communicated to the neighboring chlorophyll A, what about that deficient molecule? That deficiency is being addressed by the oxidation of hydrogen or H2O2. Oxidation of H2O2 leads to the liberation of oxygen. So over here leads to the liberation of oxygen, and oil is the key word you need to remember over here. So the loss of electron, as you can see over here. So over here loss of electron, and the next point is loss of electron, and the next point what we are going to observe is the hydrogen. So over here, though oxygen is a highly electronegative compound and oxygen is a heavily electronegative in nature, but it releases 2 of its electrons and it gives it to the photosystem 2. That's why the 2 electrons which are released from chlorophyll A are being communicated next to the stream. Okay, so this is the part where oxygen liberation is happening. So if we remember that photosynthesis results in the process formation of oxygen, this is the step at step 1 where the oxygen is being produced due to the oxidation of water. Okay, next step would be to remember that there is a hit of photon on a chlorophyll pigment, and this is the atomic structure or atomic level. Then electron which was negatively present and is close to the orbit get excited and is shown over here in a excited orbital phase. Okay, now reverting back to the image we have noticed across in the books, this is the leaf. Once the light hits, you see two electrons are being dispatched. Now these two electrons get excited, that's why they are shown with an above arrow. And now with the gradual interaction with the neighboring molecules, these two electrons, may not be necessarily the two electrons which are initiated at this point, but the neighboring molecules electrons, they start declining their energy, and during the whole process ATP formation is being noticed along with NADPH formation, and then the proton influx is present over here at step 1. So H2O gets oxidized, O2 is released in air, 2H hydrogen ions, and these 2H hydrogen ions are exactly the same which are present in thylakoid lumen. Okay, so this thing we need to remember. So sometimes teachers may ask, what is the prime source of oxygen, whether it is coming from here or whether it is coming from here in photosynthesis. So you need just to remember that in this equation, the liberation of oxygen, the liberation of 6O2 molecules is from where? So based on the colors you can judge. So O2 is actually been resulting in the release is being formulated by oxidation of water. Okay, that is the step you need to solve. This puzzle may be coined in any exams, that O2 comes from where, whether it is from CO2 or whether it is from H2O. So you need to remember that oxygen is the product which is being formed from water. Okay, and this is the carbohydrate or the glucose, water and O2 release. So again the same simple formula for photosynthesis completed with the light reaction. Next step would be of the dark reaction. Thank you very much for your time and attention.